I want you to imagine just for a second that you have a 3D blueprint for like the ultimate set of genetic scissors.
Oh.
But instead of relying on nature's incredibly slow, billion-year crawl of evolution to figure out how to actually build the thing, you hand that blueprint to an artificial intelligence.
Right, yeah.
And you basically ask that AI to just hallucinate a completely new set of building materials, like things that have never existed together on Earth, just to build it even better.
I mean, it sounds like complete sci-fi abstraction, but this is the actual reality of the research we're looking at today. We are literally taking the slow, random walk of natural evolution and, well, replacing it with directed generative biology.
Okay, let's unpack this. Today's deep dive centers on a really fascinating paper published in Science in August 2026 out of Jennifer A. Doudna's lab.
A legendary lab, obviously.
Oh, absolutely.
Yeah.
And we're looking at how these researchers successfully built these things called SynTnpBs. So these are non-natural, completely AI-designed RNA-guided nucleases that actually outperform the wild-type variants.
Which is just wild.
It is. And to get why this is such a massive leap, we should probably start with the baseline molecule here, which is TNPB.
Right, yeah. So TNPB is this tiny transposon-encoded ancestor to the famous CRISPR-Cas12 system.
Right.
It's a very minimal, multi-domain enzyme. And it uses a guide RNA, a re-RNA, to basically hunt down and cut specific DNA sequences.
And because it is so compact, it's basically the perfect scaffold to test if AI can truly design a better genetic editor.
Yeah, exactly. That compact nature is exactly why they chose it. But getting an AI to actually improve it required clearing a massive hurdle, specifically the gap between how an AI architects a protein digitally and the actual biological reality of how that protein functions in a cell.
Yeah.
So the researchers utilize a concept called inverse protein folding.
Right. And for those of you listening who might be a bit newer to the structural side of things, think of regular protein folding like reading a recipe.
Okay, I like
the
that.
recipe is the amino acid sequence, right? And you're trying to guess what the final 3D shape of the baked cake will look like. But inverse folding flips the script. You hand the AI a perfectly shaped cake pan. So in this case, the AlphaFold 2 predicted 3D structure of this specific TNPB, and you ask the AI to invent a brand new recipe that will perfectly fill that exact shape.
That's a great way to put it. But the initial attempts to do this actually highlighted a really major limitation in our current AI models.
Oh, yeah, they struggled at first.
Big time. So they started by testing a model called ligand MPNN, and it managed to reproduce the overall structural fold, which is good. But it fundamentally failed to preserve the DED catalytic triad in the Ru-VC domain.
Wait, the DED catalytic triad, that's the molecular cutting blades, right?
Exactly. The DED triad basically coordinates the metal ions that are absolutely necessary to cleave the DNA backbone.
So if you lose that triad, your genetic scissors are just, well, they're just an expensive molecular paperweight.
Yeah, precisely the problem. I mean, structurally it looked like a nucleus, but kinetically it couldn't cut anything at all. So they pivoted to a second model, which is ESM-IF1. That stands for Evolutionary Scale Modeling Inverse Folding. And ESM-IF1 actually succeeded in preserving those essential cutting blades.
But the sequences that ESM-IF1 spit out were highly divergent, right?
Very divergent.
We are talking about sharing only like 50 to 60% sequence identity with natural TNPBs. And I have to push back on the model's logic here a bit.
Go for it.
I mean, if you let an AI alter a sequence by up to 50%, doesn't it inevitably just scramble the really delicate interface residues, like where the protein has to physically grip the DNA and the RNA?
That is a highly perceptive critique, and honestly, the data backs you up completely.
Really?
Yeah. What's fascinating here is that the AI did introduce non-synonymous mutations at some incredibly crucial spots, specifically residues K76, F77, and T123.
Okay, and what did those do?
These are basically the molecular fingers responsible for recognizing the TAM, the transposon-associated motif.
Right, which is just like the PAM sequence in CRISPR-Cas9, right?
Yeah.
Little DNA barcode.
Exactly. It's a short DNA sequence that the enzyme needs to find to activate. If it can't find that TAM sequence on the target DNA, it just can't cut.
So the AI built a beautiful, structurally sound pair of scissors, but completely fused the handle shut so it couldn't actually grab the target.
Yeah, pretty much.
Why would an advanced model make such an obvious structural error, though?
Well, it comes down to a really glaring blind spot in its training environment. The AI was trained exclusively on bare monomeric proteins.
Oh, I see.
Right. It had zero context for the massive 49 kilodalton RNA DNA heteroduplex that actually docks into this 46 kilodalton protein in vivo.
Because it wasn't in the data.
Exactly. Because the model couldn't actually see the nucleic acids in its training parameters, it didn't penalize itself for mutating the residues required to bind them.
Wow. Okay, let's do a really quick recap for you listening just to lock this first part in.
Good idea.
So bullet 1, TNPB is the tiny ultra-compact ancestor of CRISPR. Bullet 2, inverse folding asks an AI to invent brand new amino acid sequences for a specific 3D shape, like filling a cake pan.
And bullet 3, the ESM-IF1 modder kept the core cutting blades intact, but it totally messed up the DNA binding fingers because it just couldn't see the DNA in its training data.
Spot on.
So an audience question for you to chew on: how would you teach an AI to respect the delicate geometry of a binding site that it fundamentally cannot even perceive?
Well, the conventional approach to that problem is brute force structural masking. You basically just mathematically freeze a radius of amino acids around the known binding site and tell the AI, hey, do not touch these.
But TNPB is so incredibly small, right? And that RNA DNA complex wraps around like almost the entire thing.
Yeah, exactly. If you freeze all those contact points, you are essentially just forcing the AI to reprint the exact wild type protein.
You'd leave it with almost 0 degrees of freedom to actually innovate, which kind of defeats the whole purpose of having an AI do generative design in the first place.
Precisely. So the Doudna lab researchers had to engineer a much more sophisticated set of evolutionary guardrails.
Okay.
They turned to a POTS model from physics.
Oh, interesting. Yeah, implementing a tool called Gremlin to analyze these vast alignments of natural TNPD sequences spanning literally millions of years of evolutionary history.
Wow.
And they were looking for 2 specific mathematical thresholds to guide the AI. The first metric is C sub I, which represents positional conservation.
Okay, let's give you a mental hook for that one. Think of the C in C sub I as classic strictness.
Oh, I like that. Classic strictness.
Right. It's quantifying how ruthlessly evolution has conserved a specific amino acid at a specific coordinate over deep time. Because if a residue hasn't mutated in 1 billion years, there is a serious biophysical reason for it, and the AI just needs to leave it alone.
That captures the essence perfectly, yeah. But conservation alone isn't enough because proteins aren't static rocks. They are highly dynamic.
Right.
So that's why they derived the second threshold, which is sigma sub I. And that measures coupling strength.
Coupling strength, okay.
Yeah, this metric identifies covariance, meaning if a specific base in the guide RNA mutates across evolutionary history, does a corresponding residue on the protein always mutate alongside it to maintain that physical interaction?
Okay. And for sigma sub I, we can think of the sigma as synergy. It's tracking the moving parts that constantly rely on each other to work.
Exactly. Synergy is a great way to remember it. So by establishing specific cutoff thresholds for both of these values, C sub 0 and sigma sub 0, the researchers constructed a really highly targeted sequence mask.
Okay, so they locked down only the residues that were either strictly conserved or possessed high synergistic coupling with the nucleic acids.
Right. Forcing those specific anchors to remain wild type. And then the AI was basically unleashed to hallucinate the rest of the structure around those locked anchors.
And did it work?
The computational results were immediate. Dual conditioning with these evolutionary parameters significantly lowered the model perplexity.
And just to clarify for everyone, in the context of these language models, a lower perplexity means the model's energy landscape is more stable, right?
Yes.
The AI is mathematically much more confident that this specific generated sequence will actually fold into the required shape and just remain thermodynamically stable.
Right, because they basically provided the model with evolutionary logic, which smoothed out all those structural contradictions it was generating before.
That is brilliant. Let's do another quick distillation of that methodology.
Go for it.
Okay, bullet 1. Freezing the whole protein was a non-starter because the AI wouldn't have room to work. Bullet 2. Instead, the researchers mined deep evolutionary data to lock down only the most critical synergistic interface points.
Yep.
And bullet 3. We used C sub I for the classic rigid spots and sigma sub I for the highly synergistic pairs.
Perfect summary.
So think about it like redesigning a classic car. What are the core components you absolutely cannot alter without destroying the core identity and function of the vehicle?
And, you know, that automotive analogy actually segues perfectly into how they validated these computational designs in the lab. They leveraged this split lobe strategy.
Oh, right. Because TNPB is a multi-domain protein structurally divided into 2 main lobes, right?
Exactly.
Here's where it gets really interesting, guys. If we extend that car analogy, you have the engine, which might tolerate some pretty radical internal redesigns as long as it still, outputs power.
Right.
But then you have the steering column. And that requires hyper-precise geometry, or the car is completely useless. For TNTB, the REC lobe is that DNA-recognizing steering column, and the NUC lobe is the RNA binding and cutting engine.
Exactly. And to find out if the AI's new designs for these lobes actually functioned, they threw them into a massive bacterial survival screen.
The Hunger Games for bacteria?
Basically, yeah. They engineered an E. coli assay featuring this highly toxic gene called CCDB. The premise is brutal, but very effective. The bacteria will absolutely die unless the AI-designed TNPB, which is delivered on a plasmid, successfully identifies, binds, and cleaves that toxic CCDB sequence.
So it is a strict binary survival output: cut the toxin or die.
Yes. But rather than testing the fully hallucinated proteins right out of the gate, they ran chimeric fusions first.
Mixing and matching.
Right. They fused AI-generated REC lobes to wild-type NUC lobes, and then vice versa. And the survival data completely validated that lobe division of labor you mentioned.
The engine versus the steering column.
Exactly. The REC lobe was incredibly fragile. Out of all the AI-generated REC lobes they tested, only a single one remained functional when fused to a wild-type NUC lobe.
Just one.
Just one. But the NUC lobe, it was highly robust. 56% of the AI-generated NUC lobes functioned perfectly with a wild-type REC lobe.
That is a stark difference in mutational tolerance. It really highlights the distinct biophysics of what those lobes are actually doing.
Definitely. I mean, the REC lobe is tasked with scanning a massive complex genome just to find a tiny, highly specific TAM sequence.
Right.
That target search requires near-perfect steric alignment. It has to be exact. The NUC lobe, however, just needs to maintain the catalytic geometry once the RNA and DNA are already docked.
So it has a much wider thermodynamic basin, allowing it to tolerate these highly divergent, wild AI amino acid sequences.
You got it. But when they finally synthesized the full models, combining the most successful REC and NUC lobe designs together, the overall hit rate was staggering. Out of 1980 entirely AI-generated designs, 466 of them, which is about 24%, showed detectable active cutting in the survival assay.
24% is huge for de novo design.
It is. But the real headline here is that approximately 8% of those active AI designs actually exceeded the cutting efficiency of the wild-type enzyme.
Wait, really? They beat nature?
Yes. That is the true inflection point of the entire paper. The AI didn't merely mimic the natural enzyme. In 8% of those variants, it successfully explored a functional sequence space that natural evolution had completely ignored, and it found a mathematically more efficient solution.
All right, pulling this together for a quick recap.
Sure.
Bullet 1, we've got TNPBs split into a precise REC lobe for hunting DNA and a highly flexible NUC lobe for cutting it. Bullet 2, the REC lobe demands rigid precision, while the NUC lobe tolerates these wild AI hallucinations.
Yep.
And bullet 3, by screening thousands of these variants through a bacterial survival test, they discovered artificial enzymes that actually beat nature's efficiency by 8%.
Perfect.
So a thought to chew on here for you listening. Does it surprise you that the part of the protein searching for the target is so vastly more fragile to mutation than the part doing the actual cutting?
It's a great question. And once you establish that kind of superior efficiency in a simple prokaryotic system like E. coli, the immediate scientific imperative is to determine if that efficiency actually translates to the vastly more complex environment of eukaryotic cells.
Right, like human cells.
Exactly. So they advanced their best-performing AI variants, which they designated SynTnpBs version 1 through 9, into HEK293T cells.
Which are human embryonic kidney cells, the classic workhorse of the lab.
Exactly. And they started by targeting a blue fluorescent protein gene, just for a really straightforward knockout assay. The wild-type enzyme achieved a respectable 28% editing efficiency.
Okay, 28%.
But the AI variants blew past that. Variant V1 hit 46%, and variant V5 hit an impressive 50% editing efficiency.
Right, that's nearly double.
It is. And they didn't rely solely on reporter genes either. They went after endogenous human loci, natural targets, right? Like RB1X1, NIBN1, EMX1, and AGBL1.
Real human genes.
Yeah. And the performance delta held up across the board. At the EMX1 locus, variant V1 delivered a 3.8-fold increase in editing efficiency over the wild type.
3.8 times better.
Yeah, and V5 showed a 3.1-fold increase. They even demonstrated cross-kingdom efficacy.
Oh, so they moved out of human cells.
Yep. They successfully edited the ATPDS3 gene in Arabidopsis plant cells, too.
Okay, I have to play devil's advocate on this data for a second.
Let's hear it.
We see higher editing rates, sure, But are these AI variants actually superior kinetic enzymes? Or did the AI just accidentally design a more highly expressed hyperstable protein that simply floods the cell? I mean, more protein equals more editing, right?
That is a vital control question. But the empirical data strictly rules out a concentration gradient as the primary driver here.
Okay, how?
They performed quantitative Western blots to actually measure the intracellular protein levels, and there was zero correlation between expression levels and the increased editing activity.
None at all.
None. Furthermore, they ran thermal stability assays. For variant V7, which is one of the most divergent structures they built, the melting temperature was 51.2 degrees Celsius.
Which is?
Virtually identical to the wild type.
Wow. So AI didn't just build a thicker, hardier hammer. It fundamentally designed a more mechanistically efficient machine.
It really did, and it managed to increase that efficiency without degrading specificity, which is always the fear.
Right, you don't want it cutting random parts of the genome.
Exactly. They utilized TN5 tagmentation profiling to systematically map out the entire landscape of off-target edits across the genome. And variant V1 maintained a wild-type-like specificity profile. It retained a strict requirement for the canonical TTG TAM sequence.
So it edits significantly faster, but it is just as rigorous about where it actually cuts.
Exactly.
Incredible. Okay, summarizing this massive leap for you guys. Bullet 1. The AI-designed variants seamlessly transition from bacterial screens to successfully editing both complex human and plant cells.
Yep.
Bullet 2. Variants V1 and V5 nearly doubled the efficiency of the natural enzyme. And bullet 3. The Western blots prove this isn't just an artifact of protein accumulation. These artificial nucleases are kinetically superior at targeting and cutting.
Spot on.
Think about the implications of that. If you had a tool that was functionally 4 times more efficient at eukaryotic DNA editing than the natural ancestors of CRISPR, where do you point it first? Agricultural resilience or targeted human therapeutics?
It's a huge debate. And to truly comprehend why these artificial enzymes possess such superior kinetics, we have to transition from the cellular data down to the atomic level.
Oh, getting into the structure. I love this.
Yeah, they utilized cryo-electron microscopy, cryo-EM, at a staggering resolution of 2.8 angstroms.
That is insane resolution.
Right. And they used it to resolve the 3D physical structure of variant V7.
And variant V7 is a wild architectural departure, right? I mean, it shares only 77% sequence identity with the wild type, and it incorporates 85 entirely AI-generated amino acids.
Exactly. So looking at it through cryo-EM isn't just about admiring the structure. It's about literally reverse engineering the AI's logic.
Okay, what did they find?
Well, if we connect this structural data back to the kinetic efficiency we were just discussing, the researchers made a phenomenal discovery. While imaging variant V7, they managed to capture a transient conformational intermediate.
A transient intermediate, like a stepping stone in the process.
Exactly. Specifically, the TAM-bound state. Yeah. This is an incredibly fleeting moment in the kinetic pathway where the enzyme has successfully recognized and bound the TAM sequence, but hasn't fully unwound the target DNA to form the R-loop yet.
And they captured that.
They captured it alongside the fully matured R-loop state, which allowed them to do a direct side-by-side structural comparison. They could literally see the atomic mechanisms that the AI invented to outperform nature.
Oh, wow. What was the first big upgrade they saw?
The first major upgrade was what we can basically call electrostatic glue.
Electrostatic glue. I like the sound of that.
Yeah, so in the NUC lobe, the AI systematically swapped out neutral amino acids in favor of positively charged arginines, specifically at positions K224R, Q227R, T228R, and K245R.
Okay, and why does that matter? Why arginine?
The biophysics of that change is just brilliant. The phosphate backbone of the guide RNA is highly negatively charged, right?
Right. Opposites attract.
Exactly. By clustering those new positive charges in the NUC lobe, the AI generated a powerful electrostatic attraction. It effectively engineered a tighter molecular grip on the nucleic acids that the wild type simply lacked.
It invented its own molecular glue.
Yes. And it also engineered what looks like a phosphate lock.
A phosphate lock?
Yeah. In the natural enzyme, there is a conserved residue, K84, which is tasked with stabilizing the very first base pair of the target DNA.
Okay.
The AI apparently calculated that this bond needed reinforcement, So it introduced an entirely new contact, N4R, to lock down that phosphate group alongside K84.
Man, while those static binding improvements are seriously impressive, I feel like the AI's mastery of the protein's dynamics is what truly stands out here.
Oh, absolutely, because to actually unwind the target DNA, the enzyme has to undergo a really complex mechanical shift, specifically a crucial kinking motion in the bridge helix.
Right.
And the AI entirely overwrote a 5-residue segment of that hinge, from Y214F through L219E.
So it replaced the structural materials completely, yet it perfectly preserved the highly conserved kinetic motion required for DNA cleavage.
Exactly.
It's like it stripped out the old biological hinges, installed completely new materials, but the door still swings with perfect millimeter tolerance.
That's a beautiful analogy.
Let's pull this final phase together for the listeners. Bullet 1. The 2.8 angstrom cryo-EM data let us actually look at the AI-designed variant V7 in pristine detail.
Yep.
Bullet 2. In doing so, we documented a brand new intermediate step in the cutting process, that TAM-bound state.
Right.
And bullet 3. We literally saw the AI optimize the system by adding positive charges to tightly grip the negative RNA and by seamlessly hallucinating completely new residues to execute the essential mechanical folding motions of the bridge helix.
Perfect summary.
It's profoundly humbling, honestly, to realize that a language architecture, fundamentally designed to just predict tokens and text, can invent a functional molecular hinge out of completely novel building blocks that just perfectly executes nature's complex atomic mechanics.
It really is. And it forces a total reevaluation of our approach to protein engineering. You know, for decades, we have operated under the assumption that we must carefully mimic the exact sequences that nature provides us.
Right.
But by integrating structural models like ESM-IF1 with the evolutionary covariance guardrails of the Gremlin pods model, this research proves we can actively design highly divergent, non-natural nucleic acid binders. And these aren't just static structures. They are dynamic machines, capable of transitioning seamlessly through complex kinetic states with even greater efficiency than the natural enzymes they were originally based on.
So what does this all actually mean? From where I sit, this deep dive is a clear indicator that the era of relying on the slow, random walk of natural evolution is closing.
I agree completely.
We are no longer limited to bioprospecting, digging through random bacterial genomes, just hoping to find a slightly better CRISPR variant. We have officially crossed the threshold into generative biology. We are expanding the designable protein space deep into de novo territory, building complex, moving biological machines essentially from scratch.
We have transitioned from discovering genetic tools to deliberately architecting them for our specific custom needs.
Which loops me right back around to the thought we started with and a final question for you listening to Mull Over today.
Let's hear it.
Nature spent 3 billion years ruthlessly optimizing these genetic scissors.
Right.
Then an AI model stepped in and improved their editing efficiency in a fraction of the time.
Right.
But we have to remember, natural evolution optimizes for survival in a highly chaotic, highly competitive environment. We are currently prompting these AI models to optimize purely for naked editing efficiency in a sterile lab setting.
That's a crucial distinction.
Yeah, exactly. So as we push these artificial enzymes to their absolute kinetic limits, what hidden constraints or secondary survival functions of nature do you think we might be inadvertently ignoring?
Oh, that is a fantastic question.
It's definitely something to think about as this field explodes. Keep diving deep, and we'll catch you on the next one.